Hi, everybody. Welcome to the first in a series of lectures on neuroanatomy. That is the anatomy of the central nervous system. I know we've spent quite a bit of time already just looking at neurophysiology, but it's a big topic, and I find it continually fascinating. So recollect from our previous experiences that the central nervous system is made up of the brain and the spinal cord. And of course, the brain has a number of different regions that we'll look at in some detail, and it connects it's connected to the spinal cord through something called the structure called the midbrain, which has a lot of complicated names associated with it. And of course, the CNS communicates with the PNS or peripheral nervous system via the spinal cord. The brain and central nervous system and spinal cord are all covered by tough fibrous membranes called the meninges. There are three layers to the meninges: the dura mater, which means tough mother, and this is where we find our venous sinuses; the arachnoid mater, or spider mother, and this is where you find the fluid-filled area filled with cerebrospinal fluid. Last but not least is the pia mater, or little mother, and this is where you find all your blood vessels that help feed the nervous system. Of course, you've probably heard of meninges before. The meninges also extend down into the spinal cord, and one of the common ways to detect infections in the brain is through doing something called a spinal tap. And one of the complicated parts of doing a spinal tap, of course, is to puncture the meninges. And I'll tell you, as somebody who's seen it done to somebody near and dear to them, it's a pretty sporty event. Of course, the common infection that people are trying to detect in the central nervous system, and something that's important to you guys as you prepare to go on to colleges, is a disease called meningitis. Of course, any itis is an inflammation. This is an inflammation of the meninges, and generally we find two common forms of meningitis, although there's others, but they are viral and bacterial. There are a number of sources for viral meningitis, and viruses are really creepy and scary and fascinating, as you guys are probably aware of, since we're recording this right during the coronavirus pandemic wave 1. So I can have a viral and bacterial meningitis, and they're both pretty dangerous. But we don't have good defenses for some forms of viruses. So often, and of course, viral meningitis will often lead to a bacterial meningitis. And remember that the nervous system was immune privileged, which isn't a good thing. It means that your body's normal humoral defenses are downregulated, that the brain's blood-brain barrier disallows the movement of large molecules into the brain. And so we are sort of limited in how we respond to disease and infection in the brain. And if you'll recollect, those glial cells called the microglia are the ones that take on the role of the macrophages in the rest of their body. But brain infections are problematic. Of course, when you have an itis, you have swelling. And when you have a swelling in the brain, you create permanent damage. Speaking of swelling in the brain, down at the bottom left, you can see a photograph of an infant with hydrocephaly. That's water on the brain. And the water on the brain that this infant is suffering from is actually cerebrospinal fluid. Again, if you recollect from our lectures in neurophysiology, two types of cells in the nervous system: the glial cells and the neurons. And the glial cells support, and they have a couple different roles. And of course, there are glial cells called the ependyma that secrete cerebrospinal fluid. Now, these ependyma line a layer of tissue called the choroid plexus, which is found inside of these ventricles. So a ventricle is a big opening. We're going to learn about ventricles when we get to the cardiovascular system as well. You have 4 ventricles. There's 2 lateral ventricles, and then there's a third and fourth ventricle as we extend down the brainstem. And the fourth ventricle is connected to the other three through a structure called the cerebral aqueduct. And the common source of hydrocephaly in an infant would be a blockage in the cerebral aqueduct. And of course, CSF will build up, and that'll cause swelling in the brain. And of course, again, swelling in the brain is bad. Remember that the cranial cavity is a pretty solid structure, and so the brain has nowhere to go, and that accumulation of fluid can damage the very soft and cheesy substance that makes up your brain. So again, the ependyma secretes cerebrospinal fluid. The cerebrospinal fluid circulates around the brain, and of course it travels down into the spinal cord. It's a clear fluid similar to blood plasma. In fact, it sort of arises through the choroid plexus as an exudate of the cerebral vascular blood supply, and it carries a lot of cool, good stuff that the brain needs, like glucose or electrolytes. So it forms it has a nutritive function, and it also protects because it's how the microglia can get around the brain. That cerebrospinal fluid carrying all those electrolytes, and electrolytes are important to brain function, allow for stable ion concentrations to be maintained. And remember that if I want to detect an inflammation in the brain or an infection in the brain, I can do something called a spinal tap, which would require me to go down into the spinal cord, get in through the meninges, and draw some of that cerebrospinal fluid out and look for things like viruses or bacteria. The spinal cord is a long length of tissue. It's about as thick around as your pinky finger, about 1 meter in length, and extends down through that foramen magnum. Remember the giant hole at the base of your skull. The spinal cord is really important for bringing information into and out of the brain. And within the spinal cord, we have arrangements of neurons that allow for spinal reflexes, which we'll look at in a little bit of detail. The organization of the spinal cord's interesting. When we talk about the brain, we'll talk about gray matter and white matter. In the brain, the gray matter is outside and white matter is inside. And in the spinal cord, the gray matter is inside and white matter is outside. And so the question is, what's gray matter and what's white matter? What's matter, really? So the spinal cord is organized into tracks, like columns of bundles of nerves, and they can be afferent or efferent. Remember that afferent ascends, and so afferent tracks are taking sensory information from the body up the spinal cord to the brain. Of course, efferent tracks carry motor information. They're sending information back out from the brain to have some sort of an effect on the rest of the body. Damage to your spinal cord is irreversible, but there's a lot of new techniques being employed to try to make repairs or compensate for that. And we're going to have a project coming up, and there's some really amazing new technologies available for people with spinal cord injuries. So the spinal cord emerges, each of the different nerve tracks. So the spinal cord gets thinner and thinner as it travels down the length of the vertebral column, because nerve tracks come out. And you can see this nice diagram that shows where all these nerves are emerging. And remember that the nerves emerge from the vertebral column through those intravertebral foramen, those holes between vertebrae, intravertebral foramen. And as those different nerves emerge out, it makes the spinal cord's diameter decrease. Of course, there's a lot of nerves at the base of the body, and so they emerge down at where we get to the sacrum and the coccyx, and they form something called the cauda equina, What does cauda equina mean? Again, here's a dissection through the gray or through the spinal cord. We've got white matter on the outside and gray matter on the inside. The gray matter is made up of neural cell bodies, right? So the soma of the neurons are in the gray matter, and then white matter, of course, is composed primarily of myelinated axons, right? Remember, myelin kind of has a white color to it just because it's full of fat. The interior of the spinal cord is organized into horns, and so we have a ventral horn, which is towards the anterior, in French means stomach, and the dorsal or posterior horns. The posterior, the dorsal horns are made up of afferent tracts, right? So they're taking sensory information up to the brain, and that makes the posterior or ventral horns composed of the efferent tracts, okay? And they are further subdivided into visceral and somatic. Remember, viscera has to do with organs. Somatic has to do with the rest of the body. So in the somatic ventral horn, I have motor neuron tracts that innervate the skeletal muscles. The visceral tracts in the posterior horn or the ventral horn are made up of neurons or axons projections from the autonomic nervous system that innervate your guts, right? You can't control them. Conversely, in the visceral posterior horn, we have axonic projections that take sensory information from the organs up to the brain, and in the last case, the somatic, the ones furthest out, take information up from the periphery of the body, your skin, your muscles, the stretch receptors in your tendons. Which brings us to reflex arcs. So you have a pretty quick reaction to noxious stimuli, like a burning candle, and you know you don't want to be burned, and the body takes advantage of the fact that it can make decisions about really bad stimulus with a collection of neurons called the reflex arc. Okay, so the most common reflex arc involves a sensory neuron, an association neuron, which leads to a motor neuron, and then we have simple reflex arcs where we just have a sensory neuron that goes right to a motor neuron and allows you to withdraw your hand from the flame, so to speak. So here's the knee-jerk reflex arc. Actually, it's not the knee-jerk reflex arc. This is a diagram of a response in the arm, but it'll be good enough. Remember, I take sensory information from the dorsal root ganglia. Ganglia is simply a collection of neurons, the dorsal root horn. So I have sensory information coming from here. In this case, I've got a pin sticking somebody's hand. In the case of the knee-jerk reflex, I tapped on the patellar tendon, and it stimulated the stretch receptors in that connective tissue, which sends a sensory signal to the sensory or afferent tracts. Now, from there, they're going to proceed up to the brain. But in the meantime, there's an interneuron here that connects it to the motor neurons in the ventral horn, which carry information directly to the muscles that will allow that knee or, in this case, arm be withdrawn. And of course, the adaptive advantage of the reflex arc is that it allows for your body to have very rapid withdrawal. And it's a very primitive reflex. It's a very primitive response in animals. Again, remember that the tracts of the spinal cord are organized into ascending and descending tracts, or the afferent and efferent tracts. The ascended tracts take sensory information up to the brain, and the efferent tracts take motor information down to the brain. And they have names like the spinothalamic tract, and of course it's going from the spine up to the thalamus, so it's an ascending tract. So I'd expect you to be able to maybe not come up with the name of the tracts, but if I gave you a name like corticospinal tract, you should be able to tell me, well, it's going from the cortex, corticospinal, to the spine. It must be a motor tract. Really pretty interesting. Pretty complicated too.